PhD student in Dr. Kristine lab.
Hi, my name is Nellie, and I'm a third-year MD PhD student co-mentored by Dr. Vivek Swarup and Dr. Kim Green. Our research focuses on understanding the role of neuroinflammation in Alzheimer's disease, and in particular, we're interested in understanding the role of microglia, which are the brain's resident immune cells in Alzheimer's disease.
APOE, or apolipoprotein E, is a protein that transports lipid and cholesterol in the brain. They are crucial for lipid metabolism and brain health. And in human, there are 3 alleles of APOE, APOE 2, 3, and 4, with APOE 2 being associated with protection against developing Alzheimer's disease, while APOE 4 is the major risk factor for developing Alzheimer's disease. If you have one copy of APOE 4, you are 3 times more likely to develop sporadic Alzheimer's disease, while if you have both allele of APOE 4, you are 15 times more likely to develop AD. So the role of APOE in Alzheimer's disease is not really fully understood yet, but it's been found that they're involved in the uptake and aggregation of amyloid beta as well as tau. Moreover, APOE actually modulates the immune response to these pathology in Alzheimer's disease.
In 2019, a case study was published by Dr. Arboleda Velasquez describing a patient who was able to escape developing early onset Alzheimer's disease by nearly 3 decades. In Colombia, South America, there is a group of individuals who very reliably all develop early onset Alzheimer's disease. The reason they develop early onset Alzheimer's disease is due to a variant in the presenilin-1 gene, and this variant reliably causes these individuals to all develop early onset Alzheimer's disease by their mid-40s. However, this 2019 case study found that there was one woman who did carry the presenilin-1 mutation and yet was able to delay the onset of Alzheimer's disease by almost 30 years. And the researchers who were studying this woman, they did genetic testing and they identified that the delay of her Alzheimer's disease was likely due to another variant that she was carrying called APOE Christchurch. And this Christchurch mutation appeared to be protecting her from developing cognitive decline until her 70s. And researchers studied this woman and they found that she had a lot of amyloid beta plaques in her brain, but she had limited tau pathology. And she also had delayed neurodegeneration and cognitive decline by almost 30 years.
So in our recent study, we wanted to model this mutation that was found in the case study in mice. So we introduced the mutation using CRISPR-Cas9 technology, and really to understand how this mutation affects both amyloid and tau pathology in Alzheimer's disease, we want to study them independently but in parallel. So we crossed them with the 5xFAD mouse model for amyloidosis and the PS19 mouse model for tauopathy. And these 2 mouse models are very well characterized and widely used in the AD community. And so with that, we found that APOE Christchurch actually reduces amyloid load in these mice. And in studying these mouse models, we found that APOE Christchurch actually reduces plaque burden in these mice, but have minimal effect on the tauopathy.
We also found that the Christchurch variant changed the cellular response depending on whether it was interacting with plaque pathology or tingle pathology. So we found that when we introduced the Christchurch variant into the 5xFAD, which is the plaque model, we found that there was an increased response in the disease-associated microglia to these plaques. However, when we introduced the Christchurch mutation to the tau model, we found that there was actually a reduced microglial response. This was an important mechanistic study that allowed us to understand how the Christchurch variant influences cellular responses to plaques and tangles in isolation. Our next goal is going to be to understand how the Christchurch variant influences cellular responses to plaques and tangles in the same animal, which is more representative of what happens in humans. We're currently working with the Model AD consortium at UCI to introduce the APOE Christchurch variant into a new next-generation mouse model of Alzheimer's disease that is carrying human amyloid beta, human tau, and human APOE4. And we hope that by doing this, we'll be able to understand how the Christchurch variant works in a more physiologically relevant mouse model, which will hopefully inform better therapeutics for treating humans with Alzheimer's disease.
This research project involved a lot of people, especially the Green Lab here at UCI and Model AD Consortium.